Welcome to this explainer. I am absolutely thrilled to have you here today. Okay, we are going to dive straight into a genuinely fascinating topic. Today we're decoding the very mechanics of cellular life. Now, biology can sometimes feel like this massive, impenetrable wall of academic jargon, right? But we are going to strip all of that away. We're focusing entirely on explaining things simply, using clear, everyday analogies to really grasp what's happening inside the invisible world of cells. Think of this as your user manual for life itself.
Here's a quick look at a roadmap for today. We'll start with seeing the microscopic world, move into how cells transport stuff, look at how they divide, and finally, wrap up with stem cells and cloning.
All right, Section 1, Seeing the Microscopic World and Translating the Invisible.
When biologists talk about the size of cells, they are dealing with numbers that are just insanely small. Take a cholera cell, for instance. It measures in at roughly 0.00015 millimeters long. Honestly, writing out all those zeros is just a recipe for confusion. So scientists use this brilliant shortcut called standard form. It's really just a way of writing very small numbers easily by keeping the main number between 1 and 10 and then multiplying it by a power of 10. So that tiny cholera cell, it's way easier to write as 1.5 times 10 to the power of -3. It's essentially scientific shorthand. Now, once we get these cells under a microscope, we need to know exactly how much we've blown them up. To figure that out, we use the magnification formula. You just take the size of the image you're looking at and divide it by the actual real-world size of the object. But here's the absolute crucial point. Before you do that math, you have to make sure your units match perfectly. You can't mix centimeters and millimeters, or your answer is going to be wildly off.
If you wanted to step into a lab and see this for yourself, prepping a slide is actually incredibly simple. You start by putting a single drop of water on a microscope slide. Then you peel off a super thin layer of an onion cell. Because cells are mostly totally clear, you add a couple of drops of iodine solution. That iodine acts like a microscopic highlighter, making all those incredible invisible inner structures suddenly pop right out in color. Finally, you carefully lower a glass cover slip on top to protect it, and boom, you're ready to explore.
Now that we've visualized the cell and we know how to measure it, it brings up a really interesting question. How does a tiny, completely sealed unit actually take in nutrients to survive, and how on earth does it expel its waste if it doesn't even have a mouth? Which leads us perfectly into Section 2, How Cells Transport Stuff, Moving with and Against the Flow.
There are basically three main ways things move in and out of a cell. Diffusion and osmosis are both passive processes. They move things from a high concentration to a low concentration, meaning they don't require any energy at all. It's literally like rolling a boulder down a hill. It just happens naturally. But active transport is a different story. It moves things from low to high concentration. And because it's pushing against that natural flow, it absolutely requires energy, specifically a molecule called ATP, to push that boulder uphill. Let's look a bit closer at osmosis. Just because it's a very specific, unique type of transport. Osmosis is entirely about the movement of water. It's water moving from a dilute area to a crowded, concentrated area through a partially permeable membrane. You can think of that membrane like a really picky bouncer at the door of a club. The bouncer is only letting water molecules pass through, basically trying to balance out the crowded party inside with the empty space outside. And animals have adapted some truly incredible ways to maximize this kind of transport. Take the axolotl, a fascinating aquatic creature. The axolotl actually relies on diffusion to breathe, getting oxygen directly from the water into its bloodstream. To do this efficiently, its gills are structured to create a massive surface area. A bigger surface area basically means way more doors for the oxygen to enter, allowing for a much faster rate of diffusion, so the axolotl gets all the oxygen it needs to survive.
Okay, so a cell can successfully feed itself, it can breathe, it can get rid of waste, but how does one single cell become a complex organism like an axolotl or a human being for that matter?
Let's move to Section 3 and figure that out. How cells divide, the multiplication game.
Cells basically divide in two primary ways, and they serve completely different purposes. First up is mitosis. Mitosis is essentially a straight copy-paste function. It produces identical cells, and your body uses this process constantly for general growth and to heal things like cuts and scrapes. But if this copy-paste function gets a mutation and starts dividing uncontrollably, that's what leads to cancer. Then we have meiosis. Meiosis doesn't want identical copies.
Instead, it scrambles the genetic code to create unique reproductive cells, or gametes, which is what ensures genetic variation in the next generation. While human cells have these complex division cycles, bacteria keep things incredibly simple. They use a process called binary fission, which is literally just growing and splitting directly in two. And they can do this fast, like sometimes every 20 minutes fast. If you ever want to know exactly how many bacteria you'll have after a certain amount of time, there's a straightforward formula. You just take the number of bacteria you started with and multiply it by 2 to the power of the number of divisions. It's pure exponential growth in action. And it totally explains why a tiny bacterial infection can make you feel so sick so incredibly fast.
Because bacteria multiply so rapidly, growing them in a school lab requires some incredibly strict safety steps. First, you have to sterilize everything, usually in a high-pressure oven called an autoclave, so you don't accidentally grow unwanted contaminating bugs. Then you pour a nutrient-rich agar jelly for them to eat and carefully spread the bacteria. But here's the most important part. You only incubate them at a mild 25 degrees Celsius. If you kept them at 37 degrees, which is human body temperature, you could easily accidentally grow dangerous pathogens that thrive inside humans.
All right, let's dive into our fourth and final section: stem cells and cloning. Blank slates. To really understand stem cells, we first have to look at the difference between generalized and specialized cells. Most cells in your body right now are specialized. They have complex structures and highly fixed jobs, like a nerve cell carrying signals or a muscle cell contracting. They can't just up and change careers. But stem cells, they are generalized cells. They are essentially blank slates. They haven't picked a career path yet, which means under the right conditions, they can differentiate and become almost any type of cell the body needs.
And this blank slate property? It leads us to a brilliant medical process called therapeutic cloning. Imagine a patient needs a tissue replacement. We can actually take the DNA nucleus from one of that patient's everyday, normal body cells and insert it into an empty donor egg. We then stimulate that egg to form an embryo, and from that we extract stem cells. Because those newly formed stem cells have the exact same DNA as the patient, we can grow them into the required tissue, and the patient's body won't reject it. It's literally perfectly personalized medicine.
It really is just wild to think about, isn't it? From simply trying to see this invisible world using standard form and basic microscopes, to understanding how cells breathe and transport water, right up to manipulating the very blank slates of life for medical cures. These simple mechanisms power absolutely all existence. Which leaves us with a pretty massive question to ponder. As our technology gets even better and more advanced, what cellular mysteries are we going to unlock next? Thank you so much for joining me in this explainer. Keep learning, and I'll catch you next time.